What's up, guys? It's Triple A Biochemistry here, and today we'll be talking about the four major types of biomolecules. There are four major types of molecules, and these are the carbohydrates, the lipids, the proteins, and the nucleic acids. Although each class contains diverse molecules, they are united under a single structural or functional definition. And today we'll be looking at talking about the first three at the molecular level because nucleic acids are special and deserve their own video. They're just so awesome that they need special attention. So buckle up, the train's moving. So we have the carbohydrates first, and simple carbohydrates or monosaccharides have the formula CH2ON, where N is greater than 3. For example, a formula for glucose is C6H12O6. If you factor a 6 out, that's CH2O with 6 on the outside. So when two monosaccharides come together, they become a disaccharide. And when they come together through a process called dehydration synthesis. Dehydration meaning water is lost, and synthesis because they come together. When they break apart, they go through a process called hydrolysis, where a molecule is added to break apart the joined disaccharide. Now for some examples. When glucose forms with another molecule of glucose, they form a disaccharide called maltose. When glucose comes together with fructose, it becomes sucrose. And when glucose comes together with galactose, it becomes lactose, the thing you find in milk. When multiple monosaccharides come together, they are also known as polysaccharides, which are the polymers, while monosaccharides are the monomers. Let's take a look at glucose again. It is sometimes convenient to draw them in this linear shape. But in aqueous solutions, they form rings. Sorry about that. They form rings. The thick line represents the one that's facing you, then the thinner line represents the side of glucose that is farther away from you. Because the carbons are so important, we like to label them, starting from the carbon to the right and going clockwise. We have one, two, three, four, five, six. These represent the positions of the carbons. Now that we've covered the structure of the carbohydrates, let's move on to the function. Carbohydrates perform essential cell functions by serving as fuel storage molecules and providing structural support. When these monosaccharides combine to form polysaccharides, the residues are linked by glycosidic linkages or glycosidic bonds, which are represented here. Cellulose is a polysaccharide that is responsible for providing structure in plants. Starch is also a polysaccharide that is responsible for providing storage in plants. Glycogen is a storage molecule that's responsible in living things and humans. The difference between glycogen and cellulose is the orientation of each glucose molecule. In glycogen, the first carbon is connected to the fourth carbon, forming what is known as an alpha 1,4 glycosidic linkage. In cellulose, however, although it is still a 1,4 carbon 1 and carbon 4 linkage here, you can see that it actually flips around. So this is what is known as a beta 1,4 glycosidic linkage because the hydroxyl group that's bonded to the carbon is above the bonded carbon. Glycogen branches out, while cellulose is more linear and it forms hydrogen bonds with each other, so that's why it's more rigid. Our second molecule is the lipid, also known as fats, also known as what I see myself in the morning every day and get disappointed in. They cannot be described into a single structural formula because they're diverse molecules. However, they do have a tendency to be poorly soluble in water because they are mostly hydrocarbons, nonpolar. Recall that water is a polar molecule because of electronegativity of oxygen, while hydrocarbons are nonpolar. So like deserves like, and polar molecules dissolve polar molecules. We can see here that the most basic unit of a lipid is the triglyceride. It is made up of a glycerol and three fatty acid chains. You get a triglyceride. Now, these fatty acid chains can either be saturated or unsaturated. Saturated if they have no double bonds in the fatty acids, and unsaturated if they do have double bonds in the fatty acid chain. Here's a cool thing. If you swap one of these fatty acids and replace it with a phosphate group, you get a phospholipid, which is the thing that makes up cell membranes. The functional lipids include long-term energy storage stored in adipose tissue, a critical component in cell membranes. The phospholipid bilayer, for example. It is also responsible for insulation and protection, which is why seals and penguins store lots of fat during the wintertime to keep warm. It is also responsible for hormone production, such as your sex hormones like testosterone and estrogen, and these are also under the class of lipids, and cell signaling, which is the thing that can prompt transcription. Next, we have the proteins. They are made up of monomers known as amino acids, and when these monomers come together, they become something known as polypeptides. There are 20 different amino acids that serve as the building blocks for proteins, which may contain many hundreds of amino acid residues. These proteins have different orders in amino acid sequence. Each amino acid, as you can see here, is made up of one amino group and one carboxyl group. The amino group is denoted as NH2, and the carboxyl group is COOH. These may be ionized in living things, meaning that NH2 can become NH3 plus when it gains a proton to bond. The same goes with hydroxyl. Then we have the R group, which varies in each amino acid. The two structures that I said, the two functional groups, I should say, that I mentioned before are always static in amino acids. They are always present. The amino group and the hydroxyl group. The R group differs. Now, when these monomers join together, they join together the same way as carbohydrates do through dehydration synthesis. However, instead of a glycosidic linkage, they are joined together by a peptide bond, as you can see here. Now, the proteins they do almost everything in cells. They provide structural support, such as actin and tubulin. They act as enzymes, such as ATP synthase. And they are also known for transcription factors, such as gene regulation. Transcription factors are things or substances that can prompt a, a gene to express itself. So it can influence the cell's behavior. And it's also responsible for transport and defense, such as your immune system. So we can see from this aquaporin here, it is transport. It transports water molecules, and those are proteins that channel across. They mediate between, they help water molecules get through the cell membrane because the cell membrane, as I said before, is made up of phospholipids and is non-polar. Water is polar, so it can't get through, but proteins like aquaporins, they can help do that. Another example is ATP synthase. It produces ATP. It is an enzyme. Enzymes are biological catalysts that can speed up reactions. ATP cannot just be produced itself, or otherwise it's going to take a long time. And ATP synthase helps with enzymatic activity and produces lots of ATPs. So we have a review. Today you learned that the four main macromolecules, the carbohydrates, the lipids, the proteins, and the nucleic acids. You also learned that monosaccharides are monomers of carbohydrates. Triglycerides are the most basic unit of lipids, and amino acids are the monomers of proteins. You also learned that the process of which two monomers get joined together is through dehydration synthesis, and how they get split apart is through hydrolysis. You also learned the structure of four macromolecules: the carbohydrates, the lipids, the proteins, and you also learned about the function of the four macromolecules. This video was made by me. This presentation was made by me. The script was made by me. The awkward pause was also made by me. If you didn't like it, you guessed it, was also made by me. If you have any questions, comments, concerns, or complaints, feel free to comment in the comment section below. If you like this video, be sure to give it a like and smash that subscribe button like you're smashing those triglycerides out of your body. It's your biochemistry professor AAA, and I'll see you next time. And next week, we'll be talking about nucleic acids. Thank you.